And it's quite a challenge for me to try to cover this in an hour and 15 minutes because I've spent 17, 18 years studying the sense of taste. It's a very exciting, fascinating field. There's so many ramifications, so many aspects of it. So what I will attempt to do today is take some highlights and share with you, and then refer you to further reading if you care to supplement it yourself. I'll try to follow the lecture outline as far as possible. I doubt that I'll get all the way through the topics on the lecture outline by the end of the hour, because I want to leave some time for slides to illustrate some of the relationships that I'll talk about in class. But before going into the sense of taste, let me call your attention to the announcements on the board. If you're interested in this seminar, I'd highly recommend it to you. This gentleman is with the California Department of Agriculture, and it would be quite interesting to see what he thinks California's future role is in the area of food. This is the information you'll need for the computer center. If you plan to use the computer center to analyze your data, especially for your analysis of variance data, I would highly recommend that you punch the cards and run it at the computer center, because it will save you hours and hours and hours of time at the calculator. So this is the information you need when you go to the computer center. If it's Greek to you, copy it down anyway, and then come and see Kathy Tassin, my technician, if you need further assistance on how to go about getting it done. She'll give you the details. You'll notice on the data that were distributed today, the data that represent what you did yesterday in class, that we have given you the total sums of squares and the sums of squares for judges for those data where we ask you to do the analysis of variance. And that's for those of you who are not ready for the computer center and wish to do it by hand. At least the hairy one is done for you, and the rest is up to you. Okay, let's look at the sense of taste. First of all, what is its function? We always like to think that, at least from a teleological point of view, that all the anatomy of the body has evolved due to some specific need and that it all has a function. Well, we are very interested in the sense of taste for a variety of reasons, and sometimes the main reason is we're interested in it because it's there, the same reason that you climb a mountain, because it is there. So perhaps the first one is simply because it is a receptor which has selectivity. And it's quite intriguing that the receptors are very sensitive to different ions and molecules, and it thereby poses a question. Is, first of all, what is the relationship between the physical, chemical properties of the molecule and the resultant sensation that we perceive? Chemists, in particular, approach the sense of taste in that way. They visualize the chemical molecule as an entity eliciting a response, and they ask, what about the molecule here is responsible for the sensation or the response here? Another reason the selectivity of the receptors of interest to scientists is that we want to ask, how does that selectivity that we call taste, how does it give rise to the pathway along the neural pathway that tells the receptor to go up to the central nervous system and say, hey, that's sweet. I mean, what is the mechanism by which this molecule travels across and gets up there and gives you a message that you in turn, from the basis of your past experience, can verbalize into a sensation? And that's the approach that a physiologist would use. A sensory physiologist wants to know, he says, okay, we have these molecules, whatever they may be. They're applied to a receptor site. They travel across a pathway. What is happening during the interim as they travel across this neural pathway? They impinge upon the higher centers of the brain, and it's interpreted as a sensation. We say, yes, that's sweet, but this is not sweet, it's salty or something else. Well, the nutritionist and the food scientist is interested in taste also because it is related in various ways to recognition of food and food-like materials, to discrimination between foods, and very closely related to acceptance and rejection. For example, without the taste receptors, we wouldn't recognize sweet substances, and maybe it would be to our benefit, since so many individuals are attempting to cut down their caloric value and they're tempted by sweet substances, but we wouldn't recognize them if we didn't have the taste receptors. We wouldn't be able to distinguish table sugar from table salt if we didn't have them to the discrimination system. And then they operate in various ways in acceptance of rejection. The culture of the individual, his environment, his nationality, his ethnic background, his entire culture has a very profound influence on acceptance and rejection, making it difficult for us to say on a generalized basis that everyone likes this or everyone likes that. They'd like to say that everyone needs milk or everyone likes milk, but acceptance rejection is greatly influenced by the culture, not just by the physiology of the taste receptor. But there is one generalization, and that is that sweet substances are avidly accepted by almost all human individuals and by many animal species. You can make that generalization. Sweet substances are highly desirable to nearly all human beings. There's some rare exceptions. And to most animal species. So the sense of taste for sweetness can serve as a reward and motivating factor, both in the human species. If you're a good boy, I'll give you some candy. If you eat your spinach, you get your dessert, that sort of reward. And with experimental animals who have been observed, for example, the rat, that they will work for a sweet stimulus. They can present them with a situation where they have to go through a maze or they have to press a bar, and their response for their reward for doing this is a sweet stimulus. So that shows that they not only discriminate and recognize it, but they prefer it and will work for it. Then the other generalization about taste is that most bitter materials are rejected. Now, you can find exceptions to that. Many of us, especially as we grow older, derive a certain pleasure out of bitter coffee, bittersweet chocolate, the bitterness of quinine water, the slight bitterness of the tannins in wines, the slight bitterness of the hops in beer. These materials are bitter, but they're in combination with other substances, and most of us, or many of us, learn to like these substances, especially as we grow older. Children are very negative against bitter materials, and it takes a while before they learn to accept things like coffee. Sometimes a child will ask for coffee because they associate it with being grown up or because they see their parents drinking it. But most children do not like bitter substances such as coffee or tea or alcoholic beverages and the like until they get a little bit older. We'll discuss how the receptors change both quantitatively and qualitatively as we do grow older. The nutritionists and food technologists may also be interested in the sense of taste because it seems to play some role in regulation of hunger, regulation of thirst, and other types of oral intake. It serves a role, but the role is poorly understood and poorly defined. And I'm sure that if all of you in this room decided to enter a research career on the sense of taste and would select a topic from any of these three areas, that you'd have more than enough to keep you busy for a whole lifetime, because there's a large amount of information that we know it must exist, but we haven't even scratched the surface. We're way back in the dark ages in understanding the senses and sensations as compared to other more technological or even other physiological aspects. One of the reasons perhaps is that the chemical senses, the sense of taste, the sense of smell, are considered less essential for survival of the human organism than are the other senses such as vision, hearing, or even touch. I presume that we could lose our sense of taste and we could lose our sense of smell, and we could still survive fairly well. In fact, we might be less discriminating and wouldn't complain about our wife's or husband's cooking because we couldn't tell the difference. But we could survive. But we would be much more handicapped if we lost our sense of sight or our sense of hearing. So from a pure survival standpoint, they are the less necessary of the senses. It's very difficult for the person entering this field for the first time or for the consumer to consider the sense of taste and the sense of smell separately. And the reason for this is that in our daily life we take things into the mouth and we perceive sensations of taste and smell simultaneously, plus everything else we perceive in the mouth. And I had hoped that yesterday's experience with sniffing versus flavor by mouth would help to illustrate to those of you in the lab the differences in sensations that you perceive when you sniff something as compared to when you taste something. And the data that the class obtained for that cookie extract and candy came out just as we had observed it to come out in previous years, just as we would have predicted, that there's quite a difference in intensity, and all of you have your own score sheets to know whether or not there were differences in quality, whether you sniff a material where the olfactory area is operating as compared to placing something in the mouth where in addition to the olfactory area you have the sense of taste, temperature, pain, touch, and saliva entering into the reaction. Now let's talk just briefly about the anatomy of the receptors. What are we talking about in terms of how we receive the sensation? You're all familiar with the normal distribution that Dr. Ressler drew on the board for you for 107A. Well, I draw the opposite one because this is the tongue. It's not the inverse of the normal distribution. All right, all over the surface of the tongue, this area here, this flat surface of the tongue is called the dorsal surface. All over the dorsal and the lateral back, near the whole area of the tongue, there are a lot of little papillae that look like dots if you look at them. They have a very rough surface, and these are called the filiform papilla. Filiform papilla. And these filiform papilla, which have a very rough surface, have no taste receptors. They're located all over the surface of the tongue, and their function seems to be to provide the roughness for the physical action of the tongue when it takes food into the mouth for mixing with saliva, and to assist in swallowing, for crushing the food against the hard palate. That seems to be the function of this rough surface. In man, that surface is not as rough as it is in other animals. If you've ever had a cow or a cat or a dog lick your tongue, you know that the filiform papilla on their tongue is very, very rough as compared to the one on your own tongue. Okay, so they have a physical function or structural function. Now there are two of the papillae on the tongue have taste functions, and the ones that are located on the lateral surface and the tip, there are a lot of them that are located in this area here of the tongue. These are called the fungiform. And the fungiform papilla do have taste buds. The fungiform papilla look like button mushrooms. If you've ever seen a button mushroom, they are shaped almost exactly like a button mushroom, something like this. If this is the surface of the tongue, if I had my tongue sticking out like that, the papilla would be in a perpendicular position to the surface of the tongue. Then the taste buds themselves are located on the lateral sides of these papilla. And the solution, this is the surface of the tongue coming like this. This is my tongue sticking out this way. You can visualize that. All right. The material that you take into the mouth, the solution flows in this direction. The solution has to come down into a moat or a depression that exists in here, and it has to impinge upon the hair-like projections or cilia of these taste buds, which have neural connections then that go via the nerve pathways up to the central nervous system that interprets the sensation and tells you whether you're tasting sugar or salt or some other substance. Okay, if you take a solution into the mouth, salt solution, sugar solution, and you don't move your tongue, you just take it into the mouth and hold it there stationary, you receive very little sensation. But if you move the tongue, then you're changing the concentration gradients, and you're caused by this moving, you're causing things to go in and out in this mode and stimulate and re-stimulate the cilia, the receptors, and then you perceive it with a much greater intensity. So movement of the tongue greatly enhances the perception of taste. This also operates in the sense of smell. Movement in the nares, where the olfactory receptors are located in the upper nasal area, movement by air is absolutely necessary before we perceive smell. I don't know whether Dr. Amrine mentioned this last time, but if you take a sniff of something and you just hold it up in there, you draw in air through the nose and hold it up in that area, you will get no smell at all until you exhale. There has to be air current movement back and forth. And that's why, of course, you hold your breath when you go past the skunk that was run over on the highway. Or if you don't like onions, you hold your breath when you drive through Vacaville. Because in holding your breath, you're reducing the air curries, the eddies of air currents that impinge upon the olfactory system. So we could say that the chemical receptors are chemical-mechanical receptors. It requires the chemical substance and it requires mechanical movement. Air movement in the case of olfaction and tongue movement in the case of taste. The other papilla of the tongue that also has taste receptors are the circumvallate papilla that are located at the base of the tongue, and there are between 5 and 12, depending on the individual. And these are called circumvallate. These have an entirely different morphological characteristic, a different structure, and I'll show you a slide of both the fungiform and the circumvallate so that you can visualize the difference between them. Now these are very visible. You can see this very readily on somebody else's tongue, and if you can hold your tongue out far enough and have a good light, you can see them on your own tongue if you look in the mirror. They're rather large structures that look something like this in the tongue. And the final set of papillae are the foliate papillae, and they're located in folds. We have some structures along the tongue that look like folds here, and these are simply called foliate. Those appear to be vestigial in man. Other species seem to have taste receptors in the foliate papillae, but in man they seem to be vestigial. So the important ones for us, as far as taste are concerned, are the fungiform and the circumvallate. The circumvallate papilla are sensitive primarily to bitter substances. And that's why it's necessary, if you take something bitter into the mouth, that you allow the material to progress clear to the base of the tongue to get the full sensation of bitterness, because these receptors are more sensitive to bitterness than the rest. All right, if we draw the tongue in terms of mapping it, in terms of the sensations that are generally perceived, we'd get something like this. For most people, this part of the tongue is sensitive to the sweet substances. Sweet substances stimulate this part of the tongue. Dr. Amrine often points out that when we eat things like a hard candy, that we manipulate the candy with the tip of the tongue so that we can stimulate more receptor area and derive the maximum pleasure and sensation from it. The salty receptors overlap, and they are primarily on the lateral surface towards the front of the tongue, approximately in this region. So these would be salty. And the sour ones, they overlap also, but they extend back further along the sides, and these would be then sour. And most of us are quite aware of a strong sour sensation that's perceived on the sides of the tongue, because the salivary glands in the cheek, the parotid gland in the cheek that secretes saliva that's very high in salivary amylase, reacts to sour stimuli. And when you get a jolt of a sour stimulus, such as if you eat a pickle or you take a bite of lemon, something very intense, these salivary glands start to secrete, and their first reaction is to contract. And this contraction is what you feel when you get a slight pain or sensation in the jaw. Some people say they feel that suddenly they have mumps because of the sour sensation. It's the gland, the salivary gland, that's contracting. But the sensations of sourness are perceived primarily here. Everyone's tongue would map out slightly differently, and this overlap is due to the fact that there are fibers in these taste buds. If they take out these fibers, they find that some of the fibers within the same taste bud will respond to sour, some will respond to sugar, some will respond to salt, so that they are not specific. You can't say this bud responds only to sour. It will respond more to sour than it does to something else, but not to sour alone. It's rather interesting that the anterior 2/3 of the tongue, about this amount of the tongue, is innervated by one nerve that goes up to the central nervous system, and the anterior 1/3 is innervated by an entirely different nerve. Now, we don't know how this evolved phylogenetically, or we're not quite certain what it means, but there's been quite a few studies of a comparative nature where they try to see, did it evolve because at one time substances in the environment which were harmful were bitter, and that is how the organism survived, by taking a small taste of things in the environment and rejecting those that were bitter because most toxic things were bitter, and then using the rest of the system to identify things as being food. Well, this is highly simplistic, and it doesn't hold water because there are many toxic materials which are not bitter. In fact, there are some toxic materials that are sweet and very pleasant, and small dosages would kill you in a short time. And there are many toxic materials that have no taste at all. And conversely, there are substances which are very desirable but can be very harmful or have no function whatever in the body. So we can't say that there's a cause and relationship between what we perceive and any survival function. We talk about taste modalities. We talk about sweet, sour, salty, and bitter. And there's still some controversy as to whether they are distinct sensations and there are distinct portions of the tongue that respond to them, or whether everything we taste is all one continuum and that we simply have points of recognition along this continuum that most of us can agree that's sweet, that's sour, that's salty, that's bitter. Now, we continue to use the classification sweet, sour, salty, and bitter, not because we think that it is infallible or that it's absolutely predictable, but because it's convenient and it gives us a working vocabulary. With most people, we can talk about sweet, sour, salty, and bitter, and they have an idea what sensations you mean. The two sensations that are the most frequently confused are sour and bitter. Those of you that participated in lab number 1 last week, perhaps some of you are among those that confuse sensations of sourness and bitterness. There is somewhat of an overlap on the tongue between the receptors that respond to sourness and those that respond to bitterness. And in addition, there seems to be among untrained people a tendency to call things that they dislike bitter, whether they are bitter compounds or not. In other words, if one goes out to the consumer and collects responses on acceptance or rejection of food, they reject something and you ask them, why did you reject it? The most frequent response you get is that it was bitter. And what they are really telling us is that they didn't like it. In other words, the term bitter at the consumer level is often meant that it's rejected, that they don't like it. It doesn't necessarily mean that it's the bitterness of caffeine, the bitterness of quinine, the bitterness of tannic acid. The early physiologists said that there were 6 basic tastes, and they included things like metallic and astringent. But later on, they concluded that what we call astringent or puckery, sometimes the layman says it's puckery, the sensation that you get in your mouth if you take in alum, which draws out the mouth, or if you pick an olive off the tree and put it in your mouth without the olive having gone through the curing process, it is very astringent. Or if you eat a green banana, it is very astringent. This is not a taste, this is a tactile sensation. The alkaloids or the tannins that are responsible for this astringency, they react with the proteins in saliva and form a precipitate on the teeth and on the mouth. Have you ever experienced that when you have this puckery sensation of astringency, that if you run your tongue along the inner portion of your teeth, you have a granulation, a coating. And this is a precipitate that results from a combination of these materials with proteins in saliva. So it's a tactile rather than a taste sensation. The metallic taste, there's still some controversy. Is it a taste or is it a tactile sensation? Most of us are familiar with the metallic taste, and sometimes it originates from compounds combining with the fillings in our teeth. We can taste the fillings in our own teeth. But there is a metallic sensation. Whether or not it's a basic taste is somewhat controversial, and I don't think that it's that important that we resolve that and keep us from making any progress. When we talk about taste qualities, sometimes by quality we can mean modality. The words can be used interchangeably. The taste quality is sweet. But even within sweet, there are differences in types of sweetness. For example, if you compare the sweetness of sucrose with the sweetness of glucose, we find that they differ not only in intensity. A 1 molar solution of sucrose is considerably sweeter than a 1 molar solution of glucose. Not only do they differ in intensity, but they differ in quality. Many individuals find that the glucose is sweet and bitter, and the sucrose is only sweet. Many individuals report that the sucrose reaches its sweetness very quickly and then drops off, so that if you were to plot a time-intensity relationship with time in seconds along this axis and intensity of sweetness along this axis, that you would have sucrose doing something like this for most people, and glucose doing something like this at the same concentration. If you raise the concentration of the glucose so that its relative sweetness is the same as the sucrose, then you might be able to have its intensity match up here, but its duration won't match up on this axis in terms of time. When we talk about the saltiness of compounds, we almost exclusively refer to the saltiness of sodium chloride, because other salts are salty and bitter, or salty and sweet, or salty and metallic, or something else besides saltiness. Sodium chloride seems to be the only compound that has a singular salty taste. Other sodium salts have multiplicity of tastes. Other chlorides have different tastes. And some chlorides, such as lithium chloride, is even toxic. So one can't exchange one salt with another. And that's one of the reasons why you seldom see anyone trying to find a substitute for sodium chloride in terms of the relative saltiness of other compounds. The one exception might be in the use of potassium chloride for individuals that are on a salt-free diet. They do obtain some saltiness from the potassium chloride, and they avoid the sodium, which their dietary might be desirable to restrict the sodium in their dietary. But potassium chloride has a different quality of saltiness. Relative to adaptation or fatigue of the taste receptors is not as prevalent as adaptation or fatigue of the olfactory receptors. If we take a strong taste solution into the mouth, we can get fatigued because of adaptation that is occurring, so that we don't perceive it as being as intense if we continue to taste and taste and taste. But we recover very rapidly. You can rinse the mouth out with distilled water, or you can eat a piece of bread or something to cleanse the mouth, and you return back to your original sensitivity. It takes much longer to do this with olfactory stimuli. Perhaps many of you were fatigued yesterday after sniffing so many compounds. You began to feel not necessarily a pain in the nasal area, there's probably a pain in the back, but you felt like you were really utilizing your senses almost to exhaustion or to fatigue. fatigue standpoint. With experience and with exposure, the individual learns to perform for longer periods of time and to recover much more quickly. So there is a learning that occurs. With the professional tasters of alcoholic beverages or coffee or tea, or the professional perfumers who do a lot of sniffing and compounding of perfumes, they recover very quickly. And they have sensitivity of a long duration. Part of it is motivation, part of it is experience, and part of it is simply that they have learned some ways of minimizing fatigue or adaptation. With tasting, with food products, one of the ways of minimizing the fatigue or adaptation is not to swallow the substance. And that is why the professional tasters do not swallow. A certain amount goes down the throat and is swallowed anyway, a few tenths of a milliliter. You can't help it because of the swallowing reflex, so a certain amount always does go down. But professional tasters do not consume the items that they're actually evaluating. When Dr. Amering was talking about odor, did he mention odor thresholds in any detail? Did he comment on them? I have some personal feelings about the value of thresholds. I'm a little concerned when I read the literature and I find that chemists in particular are measuring the thresholds to various odors and to various taste compounds, and they present table after table of information in terms of milliliters per liter or so many molecules per volume of air for thresholds of different compounds. And the reason this bothers me is because without information on what happens at the concentration that the material occurs in the normal state, we can't use the threshold for predictive value. Let me draw a little graph to show what I mean. If we have concentration of the compound, whether it be a taste compound or an odor compound, and we have the response here, we may find that the threshold for compound A is at this concentration. And let's say that we were using a paired comparison technique where you had a blank versus the experimental sample that has the concentration in it. So you have a blank versus a concentration. In a paired comparison, you could get them correct due to chance alone 50% of the time. So by definition, then the threshold would be the 50% level. Okay, so the level at which it was detected half of the time, that could be called the threshold, that could be called the 50% threshold, and maybe compound A has that threshold. Let's assume that the compound that we're tasting or sniffing has that threshold. Then you try compound B, and you find that it has this threshold here. So you conclude then that because you have a lower threshold for A, that A is much more important in the food in which it occurs as compared to B. Isn't that sort of why they do threshold values in terms of the basic research with taste and olfaction, to see what the relative comparison would be? Well, my personal opinion is that having one point on a continuum tells you nothing about where it's going to be if you get up to the concentration at which it occurs in the food. Say that A and B occur in the food at this concentration up in here. Say it occurs at this concentration. You may find that by increasing the concentration and sniffing or testing the material, that you get a progression that looks like this for A, and one that looks like this for B. So that when you get out here to that concentration at which it occurs in the food or beverage, that A is no longer the more intense or the more important, but B is. Does that sort of explain why I feel that basing sensory information on thresholds can be very incomplete and might give you an erroneous bit of information if you don't continue to test it at higher concentrations. Now, it's rather interesting to look at the literature and find that it's the chemists who are reporting thresholds, not the people in sensory analysis. It's the chemists. And I think the reason for it is because it's easy for them to comprehend and visualize and work with things that they can express in physical terms, such as molarity or normality or percent or parts per billion or whatever their units are, because it gives them something they can sink their teeth on. They know what they're talking about. But if you get out into this region of intensity evaluations, then you have to get into statistical analysis, and then you have to use probabilities, and perhaps they feel a little less secure when they get up into this area. But if we're going to predict what's happening in a food from thresholds, then we have to have this information as well, not just this. I sometimes say that thresholds are convenient statistical endpoints for chemists. Relative to pleasantness and unpleasantness of tastes, those of you that were in the lab number 1, you may recall that I showed a graph of data that we collected, responses of people to the taste of sodium chloride solutions and sucrose solutions, and we found that as we increased the concentration of either salt or of sugar in solution, that 1/3 of the people said the more salt it had, the better they liked it. 1/3 said the more salt, the less they liked it, and the other third said they liked it up to a certain concentration, after which they didn't like it anymore. So if we try to make some generalization about what is the influence of the concentration of the taste solution on pleasantness, we would be in trouble to try to generalize if this is the dislike part of the scale and this is the like. We get one group that does this, one group that does that, another group that does this in the studies that we have conducted and that have been reported in the literature as well. So I think the lesson to learn from this is that if you're working with degree of like and dislike, that you will have to clearly define your criteria so that every judge has a correct frame of reference. And by a correct frame of reference, I mean a consistent one. One should ask him questions relative to like and dislike for practical things. If you give him a wine and ask him if he likes or dislikes it, that's what I call practical. But if you give him a salt solution in 50 milliliter beakers under red lights in the laboratory and ask him whether he likes it or dislikes it, then I feel that you've taken it out of context and you haven't defined what your frame of reference is. Does he like it or dislike it for what? For gargling? For his drinking water? To mix with his tequila? I mean, what is your frame of reference? So I think in terms of likes and dislikes, these should be placed into the proper context of a practical situation. I think it's meaningful to ask if you like and dislike a food, but rather impractical to ask if you like and dislike a solution of citric acid or acetic acid. This particular graph also illustrates the next point, number 8, the effect of concentration. You may find this concentration of sugar very desirable and like it, but you may find that as it gets higher it becomes too sweet and you dislike it. So when one talks about like and dislike, we have to take into consideration what the concentration is. And we have to recognize, too, that all of us fall on different points of a continuum. We can have a normal distribution for the average, but we as individuals may fall outside of the average and be over in the tails of the normal distribution. I won't go into taste and chemical configuration because it would take too long to try to tell you about the various theories that have been proposed to explain why a compound of a given configuration or with a given electrical charge is thought to induce a certain sensation. Many people are working on theories of taste and olfaction, but so far no one has proposed a theory that answers all the questions of what we perceive behaviorally. They can't explain many of the sensations and the behavioral patterns that humans perceive and respond to. I will mention that there's two types of taste theories. I'll mention them by name, because you might read about them, or a friend might ask you, well, how do we perceive taste? Well, there's two theories that are proposed, and I'd like to mention them just by name. One is called a specificity theory. And the other is called a patterning theory. Two main theories of taste. And what they say is the specificity theory says that we have a specific receptor that responds to a specific taste. We have a receptor that responds to the sucrose molecule, or we have a receptor that responds to the sodium chloride molecule. That's a specificity theory. Well, more people subscribe to the patterning theory than subscribe to the specificity theory. The patterning theory says that all receptors are alike and that they respond to all taste stimuli, but not equally so. All receptors are alike and that they respond to all taste stimuli, but not equally so. So one receptor, if this were a receptor, it might respond to this degree to sweet, it might respond to this degree to salty, to this degree to bitter, and to this degree for sour. So what one perceives primarily is salty, but that doesn't mean that the others are not operating. So this would be sort of a patterning theory. And it appears on the basis of animal experimentation with actual nerve fibers that this is, in fact, what happens, at least with animals. Some testing has been done with human subjects, and that's rather intriguing just because of the uniqueness of the situation. There are many individuals, particularly older individuals, who have hearing difficulties, and the only way to improve their hearing, to alleviate their hearing problems, is to immobilize the stapes, one of the little bones in the inner ear. So they go in through the ear canal and immobilize the stapes so that little bone doesn't vibrate and interfere with the hearing in this hearing loss that the individuals are perceiving. Now, when they go in to the inner ear, the chorda tympani nerve that goes from the anterior two-thirds of the tongue back to the central nervous system, it crosses right across that ear canal. So when they go in for that operation, they have to move that nerve over to get to the stapes. So investigators in Sweden, Zauderman and his co-workers in Stockholm, were among the first to perform this operation and this test. They get the permission of the patient in advance. And they ask him, is it all right, as long as we're in the inner ear, to perform some very simple experiments? We'll flow some solutions over your tongue while you're anesthetized, and we'll place an electrode on the chorda tympani nerve, which is going to be exposed in your inner ear, and we'll measure the responses or the impulses that travel across it. So they have obtained the permission from maybe 20 or 30 different people who have had this operation. And they have been able to determine then the pattern of the electrical response that goes from the tongue up to the central nervous system. And they have found, for example, that there is a very direct relationship between the sensitivity of the person before he was anesthetized. They give him some behavioral tests like some of the ones you've had. They measure his sensitivity to sweet, sour, salty, and bitter compounds. Then when he's going through this operation, he's anesthetized, they flow over his tongue sweet, sour, salty, and bitter solutions, and they find that the magnitude of the neural response is equivalent to the magnitude of the behavioral response for that individual patient. Now this sounds very simplistic because they've been doing this for animals for dozens of years, but it's only recently that they have reported this for human subjects. Many animals respond specifically to the taste of water. So for many animals, it's not only what we would call sweet, sour, salty, and bitter, but there is a definite response to water. But man does not. Man does not give a specific response to water when water is flowed over the tongue. The flowing of water over the tongue results in a response in the tactile receptors and in the thermal receptors, but not in the taste receptors. But in many species, several species of birds, for example, the dog and the cat, they find that there is a taste receptor that responds to water. And perhaps there are, this is distilled water, so it's not due to some anions that might be present there. Some people are unable to taste anything at all, and that situation of not being able to taste is called agousia, the inability to taste. That is analogous to anosmia. The anosmia is the inability to smell. And the inability to taste, usually this is due to an injury that the individual may have sustained, a blow to the head, for example, or if during surgery the taste nerve was cut say that while they were immobilizing the stapes of the people that were having the inner ear operation, they inadvertently severed the chorda tympani. Then on that side of the tongue, the individual would no longer be able to perceive sweet, sour, and salty stimuli, and he would be agousic on that side of the tongue. Sometimes individuals have a nervous disorder, either due to injury, a blow to the head, or to a disease that affected the nervous system, where they taste only one thing and everything tastes like that. In other words, everything they taste tastes bitter, or everything they taste tastes putrid, or some other very unpleasant thing. Most of those studies reported in the literature are very sad cases, because the patient comes in and says, everything that I taste tastes like dirt, or tastes like rotten meat, or something very unpleasant. And they really have they're really bad shape. You can imagine how it would affect their entire life if everything they taste was unpleasant this way. And they find that it's some disorder, sometimes called paragousia or hypogousia, that is influencing this. But it's very, very rare. All of us differ in our sensitivity. Some of us are more sensitive to one thing than to another. I know for myself that I'm not very sensitive to low concentrations of sweet substances or low concentrations of bitter substances. But I'm very sensitive to salt and extremely sensitive to sour substances. And this is in no way reflected in degree of liking, that I would like one type of thing more than another. But I know from my own daily experience in sensory analysis that I'm not good at all when it comes to low concentrations of sweet or bitter materials. So when we work with bitter substances and sweet substances in the lab, I never select the concentrations to use. I rely on the judgment of other people whom I know have better, more developed senses in those two categories. But I feel confident in selecting sour and salty materials when it comes to setting up things for a laboratory experiment. One of the biggest mistakes one can make, of course, is to rely upon his own judgment as a criteria for selection of a concentration to use, and then find out that everybody else is gagging on the concentration or doesn't perceive it at all. The effect of temperature on taste, I think we could make a generalization that would once again look like one of these normal distribution curves. I can leave this one here. If I change this to temperature and leave this as sensation, if we chill a solution, we reduce the ability to taste it. Lowering the temperature of any sweet, sour, salty, or bitter material will reduce the ability of the judge to taste it. Elevating the temperature beyond some value here, maybe 40 degrees centigrade, reduces the ability to taste the sensation also, so that we have maximum ability to taste, depending upon the material, somewhere between 10 and maybe 30, 35 degrees centigrade. Body temperature is 37, as you recall. This is not necessarily true for olfaction. This is true only for taste. But it seems to be true for all of the four taste modalities. The effect of the taste medium, what do we mean by the taste medium? The form in which the taste is presented to you. One very dramatic way of illustrating the effect of the taste medium is, have any of you ever taken rock candy? Rock candy is just about 100% sugar. If you take rock candy in the mouth, if you like sweet things, you'll find it pleasant. But if you take a 40 or 50% sucrose solution into the mouth, you'll find it very unpleasant. It's sickening sweet. All right, why is 50% sucrose in solution sickening sweet, yet 100% sucrose in rock candy pleasant? Does anyone want to speculate?
When it dissolves slowly in the saliva.
That's part of the answer. You're correct. Part of the answer is the rate at which it dissolves in the saliva. If you take a solution of 50% sucrose into the mouth, you can't secrete enough saliva fast enough to dilute it to a concentration that is pleasant. But if you have rock candy in the mouth, you're not perceiving 100% sucrose. You're perceiving that amount that dissolves in your saliva. And some very interesting experiments have been conducted using both rock candy versus solutions and rock salt versus salt solutions. And they have asked individuals to take the rock salt or the rock candy, manipulate it in the mouth until they're just about ready to swallow the resultant saliva. But instead of swallowing the saliva, they expectorate it and they measure the concentration of the salt or the concentration of the sugar in the saliva. And for sugar, they find that on the average, people swallow when the concentration of the saliva is between 15 and 25% sugar. Then when they take these same people and they ask them to express a degree of liking for a concentration series of sugar, they find that those whose saliva is 15% prefer a solution of sucrose that is approximately 15%, and those that have a higher amount in their saliva prefer a sucrose solution that is approximately 25%. So it appears then, on the basis of some very small number of experiments, that the person swallows the saliva when it reaches a concentration that they find pleasant. Another effect of the taste medium, you saw this for the odor medium yesterday. If you dissolve the taste material or the odor material in water, you would get one type of sensation as compared to dissolving it in alcohol or in oil. If it were in a solid compared to being in a semi-solid, all of these things influence the rate at which the substance is liberated and the time period or the duration that we perceive it. Often we're asked, well, what is the effect of age? The differences between children, the differences between adults, the differences between adults of different ages. Well, just from an anatomical point of view, the anatomists and physiologists have recorded the number of taste buds, and the newborn infants have a much larger number of taste buds than do children, and children have more than adults, and middle-aged adults have many more than old adults. So it appears that we lose taste buds as we grow older. There's fewer and fewer taste buds. This does not necessarily mean that your ability to taste is diminished, but you have fewer taste buds operating. Just as an example, in one study of a large population, when they examined the taste buds of children between the ages of 1 year and 6 years of age, they found that the children had an average of 248 taste buds per papilla. For each of these papilla, 248 taste buds in each papilla. Then they examined a group of patients in a nursing home, and the patients were between the ages of 75 and 85 years of age, and they found that on the average, those people had 88 taste buds per papilla. And not only were there fewer taste buds, but many of these 88 taste buds were atrophied. They were sort of non-functional. I'll show you a slide a little later that shows that the ability to taste and the ability to smell does diminish somewhat with age. One can counteract this through experience, because one can learn a great deal about perception by simply being highly motivated and having a lot of experience. Relative to differences in taste ability between men and women, there's a lot of controversy in that area, but in general, it appears that women are slightly more sensitive, especially to bitter and sour sensations. Recently, we have been involved in an interlaboratory study, 4 laboratories in the world, one in Sweden, one in Poland, one in the Netherlands, and our laboratory here in Davis. We each tested 25 male and 25 female students, and then compared our results. And we found that in all 4 laboratories, the females were significantly more sensitive to the taste of sodium chloride and to the taste of sucrose in solution than were the males. Now we're talking about a small population. There were 25 males and 25 females in each of 4 laboratories in these 4 parts of the world. And the material we tested was sodium chloride in solution and sucrose in solution. But all 4 laboratories got the same results in the same direction of the response. So this makes you begin to think that it might have some validity. Smoking, there's a great deal of controversy on the effect of smoking. We have studied the effect of smoking, and we have only added to the confusion and the controversy, because although we find that in general the non-smokers are slightly more sensitive, even with large populations and large replications, we don't get the difference to be significantly so. I'll show you a slide on that. Time of day, it's not so much the time of day as it is the relation to meals and the relation to fatigue. Usually one is more sensitive between the hours of 9 and 11 in the morning and 2 and 4 in the afternoon. And I don't think it's because of the time of day, but I think it's because of it's long enough after breakfast and long enough after lunch and not so close to lunch and not so close to the evening meal that you have things like hunger entering in. And also ability to taste or smell is related to how attentive you are, and you're less attentive when you're tired. So these diurnal or time of day variations are interrelated with meal patterns and with fatigue. Well, I'd like to show some slides. I think they will illustrate what we've talked about and sort of finish up most of the rest of our outline here. This is the surface of the tongue of a puppy dog. I think some of you have seen this slide before. This is the surface of the tongue. These are the filiform papilla that are rough, that seem to have more of a support or a roughness function rather than a taste function. And this is a fungiform papilla that I told you was mushroom shaped. It looks like a button mushroom. All right, it sticks out on the surface of the tongue, and the taste cells, the taste receptors, are down here in the moat. And you can see how movement of the tongue would assist in getting taste solutions into that moat to stimulate the taste receptors. Now, did that go forward or back? How about this? Forward. This is the surface of the tongue of a human baby. And these now are the supporting or filiform papilla. And see how smooth they are as compared to the tongue of the puppy dog. And that's why ours are not as rough. And this is the fungiform papilla that is the mushroom-shaped one. These photographs were taken with an electron microscope. That's why you have the obvious depth of perception. The taste receptors are located in this moat, and they face out perpendicular. This is perpendicular to the tongue, but the receptors themselves are parallel. This is an electron micrograph of the circumvallate papilla at the base of the tongue that forms a chevron. And you see their structure is entirely different. They aren't mushroom-shaped at all. They have the outer portion that you see here, and then this inner portion, and the taste receptors are in the moat. And it requires manipulation of the tongue to change your concentration gradient and get the sensation at maximum there. Forward. These are data that are obtained with rats that show how important sweet sensations are to the rat, that the rat will work for it. The data along this line here are rats whose taste nerves have been cut so that they have no ability to taste. They've been desensitized. And along this axis there is a increment of how many times the rat will press a bar to get a sweet reward. So the rats that are normal have normal taste sensation. This is the frequency with which they will press a bar to get the reward of sodium saccharin, which is sweet to man, as the concentration of saccharin grows up. Then we see here, as the concentration of sucrose is increased, how the rat really has a great time here and presses the bar. He just sits there at the bar and presses it continuously because he gets such a charge out of the sweet sensation from sucrose. These rats, on the other hand, have had the taste nerve severed, and although they do show some response with increasing concentration, it's certainly not the magnitude of those whose taste ability is intact. The fact that they do show some response here suggests that perhaps there's some clue that they get. Maybe it's the viscosity, maybe there's an odor, maybe there's some sort of a tactile sensation that they perceive that they find desirable. These are the data I mentioned to you previously. The effect of increasing the amount of salt on whether one likes or dislikes the solution. We found here with testing the small number of people that are listed there at the top that some people say the saltier it is, the more they like it. Others say the opposite, and a third group is in between. The same is true for sucrose. Now, we made the mistake of only going as high as an 8% sucrose solution. It's very obviously sweet, but it isn't overwhelmingly so. And it's highly possible that if we had continued this out to, say, 20% or 25% sucrose, that everybody would eventually have reached a maximum of life and would have plateaued off. So it is possible that what we see here are various portions of a somewhat normal distribution curve, with this one peaking here, and this one may have peaked somewhere way back here, and this one would have peaked somewhere up there. Because surely if we had continued out here indefinitely, people wouldn't have continued to say they like it and like it and like it. I'm sure at some point in time they would have reached a maximum and then dropped off. Another thing you notice is this is a distilled water blank. And people did not start at the same place, did they? In terms of liking, these people perhaps are saying that they don't like distilled water and that adding sugar makes it taste better. And maybe these people are telling me that they like distilled water and adding the sugar makes it taste bad.
Was the distilled water sample mixed in with the sucrose sample?
Yes, all these concentrations were dispersed in distilled
So
water.
it might, when you take some distilled water, it's hard to tell, right after sweet.
It's possible these were all presented in a randomized order so that the order effect was counterbalanced across the whole series. And maybe that's what these people are saying here. They don't like distilled water and salt makes it taste better, and vice versa. I do want to point out is that the people who are represented in this distribution are not the same individuals that are downward here. In other words, the pattern here is independent of the pattern there. If you like sucrose, it doesn't necessarily mean you'll dislike sodium chloride or vice versa. They're not interrelated. Okay, this is a very interesting study that was conducted in England among a large number of patients in a hospital where they measured their blood glucose level. Then they gave them solutions of sucrose. They gave them a distilled water solution. They gave them a 5% solution, a 10%, 20%, and 30% sucrose solution and asked them which one they liked best. And they found that when blood glucose level was below normal, normal is 50 milligrams per 100 milliliter, when it was below normal, that the people preferred 30% sucrose solutions over the other alternatives. When it was higher than normal, they did not prefer the 30% sucrose solution. So when plotted, it forms this line. Now, this study is quite interesting because when one reads it, it appears to have been a very valid study with a large number of subjects and conducted under good control. Yet no one has ever been able to duplicate this, either with experimental animals that have been rendered hyperglycemic on purpose through insulin administration, or with other subjects whose blood glucose levels have been altered or who have differing blood glucose levels. So to my knowledge, this study has never been duplicated. So it makes you wonder whether it was an isolated fluke or whether we simply haven't been able to duplicate the procedure to get this type of result. But anyway, there is a suggestion, in this case a very dramatic one, that our own blood glucose level influences our response to a sugar solution or a sugar sweet material. Here's the influence of age on perception of taste. This work was also conducted in England among a very large population of medical students who are between the ages of 20 and 29, and then in a rest home among older people, and they subdivided them into groups as indicated here. The taste response that they used was to apply electrical current on the tongue and measure how much electrical current was necessary before they perceived a taste sensation. They didn't use taste solutions. Okay, so these are expressed in microamps, and you see just the relative difference that for the people, the students, it took only 10 microamps to produce a taste sensation, but it takes 30 to 50 microamps, and it increases as the people become older. The sense of taste, just on the basis of the amount of electricity necessary to produce a sensation on the tongue. We can express this another way if we take these data that were reported by Dr. A. Moore, the same gentleman who gave the seminar yesterday. He reported these data for the people that he tested in their responses to the odors that he was measuring. He had the age groups as indicated here, and this is the relative sensitivity. And he finds then that people differ considerably. We have an older individual who is above average here, and we have younger individuals who are below average there, but the regression line follows a downward trend, saying that the older the individuals he tested, the less